All right, it's my pleasure today to continue our Psychopharm Friday interview series with a special guest. This is Dr. Terry Robinson. He's the Elliott S. Valenstein Distinguished University Professor of Psychology and Neuroscience at the University of Michigan. He is probably the person who I've interviewed who has the most papers in psychopharmacology, of people I've talked to at least, with almost 20 of them. And they span really a very large range of the modern history of the field over about 35 years, with his first paper being in psychopharmacology being in 1984, and what I think he's described as his last experimental paper, led by him at least, in the journal in 2020. And intriguingly, both of these papers really have some very similar themes. So I think it would be really nice to hear him discuss them kind of as bookends of a story, a scientific story, as well as in some ways a scientific career. So first, I would, of course, just welcome Dr. Robinson. Thank you for being here.
Well, thanks for having me, Steve. It's a pleasure to get a chance to chat with you.
Yeah, my pleasure entirely. Okay, so let's start with his paper about 30 some years ago now, from 1984, which was actually a single authored paper by Dr. Robinson. It was entitled Behavioral Sensitization, Characterization of Enduring Changes in Rotational Behavior Produced by Intermittent Injections of Amphetamine in Male and Female Rats. So Dr. Robinson, could you tell us a little bit about the main questions that you asked in this paper and some of the main findings?
Yeah, sure. I mean, back then, sensitization was a relatively new phenomena. It had been described much earlier, but there weren't a lot of papers on it. A lot of papers on the repeated effect of particularly psychostimulant drugs, focused on tolerance. And so this idea of sensitization, that is, rather than a decrease in a drug effect with repeated drug administration, sensitization is an increase in a drug effect upon repeated administration, was just starting to get some attention in the field. And it hadn't been really well characterized in terms of what kinds of conditions are best produce sensitization, how long does it last, are there sex differences, and so on and so on. And so this paper was really just an attempt to characterize sort of some of the major features of the phenomenon of psychomotor sensitization. Again, many different drug effects can sensitize. One class of drug effects that sensitize are their psychomotor activating effects, the effects that you usually think of associated with giving amphetamine or cocaine. Animals get wired, run around a lot, and so on. And so we were looking specifically at psychomotor sensitization, and a lot of the studies at that time were on psychomotor activating effects, I think for a couple of reasons. One, we knew a lot about the neural systems that mediated psychomotor activating effects, and they involved dopamine systems. So it gave sort of an insight in terms of what was happening in dopamine neurotransmission. And two, it's relatively easy to measure relative to a lot of other things one might measure. Now, in this experiment, we actually measured rotational behavior. And this is rotational behavior in rats that have a unilateral 6-hydroxydopamine lesion of the dopamine system. And the reason for using rotational behavior, and we did a lot of studies back then with rotational behavior, was because if you look at something like locomotion, there's a very sharp inverted U dose effect curve. That is, from the dose that initially produces locomotor hyperactivity, you only can go up, a 2 or 3-fold at the most before locomotion goes down, because the animals are going into stereotypy. And so, sometimes down is up and up is down when you're trying to use something like locomotion. So it can be really easy to miss sensitization effects with locomotion. With rotational behavior, on the other hand, you have a very wide linear dose effect function where up is up is up is up. And so it makes it much, much easier to quantify the degree of sensitization, which is very difficult to quantify degree of sensitization with something like locomotion. So we just did a number of things, like we compared, do you get sensitization with a single injection? Is it bigger with multiple injections? With the single injection, we did things like withdrawal for different periods of time. I think it was like 1 day, 5 days, 30 days, and 12 weeks or something like that. That is the time interval between injections. Does that influence the degree of sensitization? We looked at things like sex differences. We looked at whether conditioning could account for it. in that experiment. We did a cross-sensitization study, that is, do you see sensitization to another drug? And so we characterized that. And some of the main features of sensitization that were identified in that study, which many other people have reported as well, is that you tend to get greater sensitization with widely spaced or intermittent injections than you do with injections that are close together in time. And that's like a real hallmark of sensitization. We found that it was extremely long lasting. Even after a single injection, the animals were still sensitized up to 12 weeks later. There were sex differences. Females tend to sensitize more readily than males, and something else that's been reported by a number of people, and so on. So it really just sort of was getting the lay of the land and characterizing the phenomena, which, you know, again, hadn't been that well characterized at the time. So that's 1984.
Yeah, some foundational work.
One other thing I'll mention about these early sensitization studies in terms, and also this one, if you read those papers, addiction is never mentioned. That is, none of these early, in the 1980s, I probably published 20 papers on sensitization, and not in a single paper is addiction mentioned. Sensitization is all discussed in those early papers in terms of an animal model of amphetamine psychosis, not addiction. We didn't make the connection to addiction until the 1993 paper with Kent, you know, which was the incentive sensitization theory paper. But lots of sensitization studies, no addiction mentioned.
Interesting. Yeah, so this is one of those cases where you're sort of studying a phenomenon for one reason and ends up maybe being really relevant to, you know, I think probably the theory is most famous now in the context of addiction. And so it's kind of one of those the stories where you started going in one direction and ended up finding that it's, you know, there's more fruit, so to speak, in another. That's an interesting story that I think comes up a lot in scientific careers as well. And then one other thing I thought was interesting about that paper, especially for the time, was that you really spent some time looking at sex differences and also hormonal differences that might influence this phenomenon of psychomotor sensitization. Can you comment on whether that's true, that was unusual for the time? My impression, you know, was that most of these studies have been done in males over the years, and yet here's this paper that really dives into this in the sort of early first half of the 80s. Was that an outlier, or is it something that you know?
No, there's an actual interesting personal reason why we were looking at sex differences to some extent. A lot of these early papers with Jill Becker, and Jill is very well known for looking at sex differences. And even before this 1984 paper, Jill and I had published a number of papers looking at sex differences in terms of psychomotor effects of drugs and so on. And so Jill's influence was such that, yeah, we were going to look at sex differences. And so looking at sex differences, I'm sure, was primarily due to her influence on these early studies. And of course, she has continued for her entire career looking at sex differences and the effects of gonadal hormones on a variety of different drug effects.
Sure, yeah, and I think this is a topic that really, I think, many of us are sort of forced to consider now, whether we like it or not, because it's become so apparent due to work by people like Jill and Dr. Becker and your many others, just showing that this pops up all the time. And I probably will get back to this topic too, but I thought now would be a good time to kind of fast forward about 35 years to your 2020 paper. This is Carr et al., Carr, Ferrario, and Carrie Ferrario and yourself. Intermittent access cocaine self-administration produces psychomotor sensitization, effects of withdrawal, sex, and cross-sensitization. So could you tell us a little bit about what you found in that paper and how that informed our view of this topic more generally?
Yeah. So this is a paper that was done by Crystal Carr, who was my last graduate student. And the issue there is there's been a controversy over the years in terms of what kinds of drug regimens are most effective in producing sensitization. Almost all of the early studies, of course, were with experimenter administered drug. And especially when you get into the addiction connection, that can be criticized pretty easily because people don't get experimenter administered drug, they take it themselves, they self-administer drug. And there's been a controversy over the years as to whether or not self-administered drug, like experimenter administered drug, produce things like psychomotor sensitization, dopamine sensitization, incentive sensitization, and so on. And there were a number of early studies by a number of people that showed that self-administered amphetamine or cocaine did indeed produce psychomotor sensitization. But all those early self-administration studies involved what these days might be called short access self-administration, where animals were given access to drugs for 1, 2, maybe 3 hours a day. And short access, of course, has been criticized as not really being a model of addiction. And Med and Koob, published a very well-known paper back in the 90s, suggesting that to really produce addiction-like symptoms in animal models required what they called long access, which was giving animals, particularly with cocaine, access to cocaine for, up to 6 hours plus a day. And it's true that is, if you compare short access with long access produces a number of behavioral changes that are addiction-like. And so long access for many years now is touted as, what you need to do if you really want to study the kind of changes in brain that take place by drugs that lead to addiction. And a number of people pointed out that when you look at animals exposed to long access, particularly cocaine, if you look at the animals early after withdrawal from self-administration, you tend not to see sensitization. That is, they tend not to show psychomotor sensitization. There's reports where you see reduced dopamine release on a drug challenge rather than enhanced dopamine release, which is associated with psychomotor sensitization, and so on. And so the idea was that fits in with the idea that, well, in addiction, there's really a hypodopaminergic state, and these long access self-administration studies supported this sort of notion of a hypodopaminergic state in addiction, and that the reason people take drugs is to overcome this so-called dopamine deficiency. However, more recently, there are a number of people who have started to look at what's called intermittent access self-administration. And intermittent access is thought to better reflect the pattern of drug use, particularly cocaine, and most of the studies are with cocaine, but not all of them, where you get spiking levels of cocaine in brain. And this model was developed by a graduate student of Dave Roberts, Ben Zimmer. And I actually remember talking with Dave at a Society for Neuroscience meeting where they had a poster on this intermittent access stuff. And what they were showing was intermittent access produced much greater addiction-like behavior than long access, even though the amount of drug intake was like short access. And you had this intermittency. Well, we've known for decades that intermittency is critical in producing sensitization. That got us to start to look at intermittent access. And indeed, if you look at animals with intermittent access, you get greater addiction-like behavior than you do with short access, but also greater addiction-like behavior than you get with long access, even though the levels of drug intake are much, much, much lower. And so we've reported incentive sensitization with intermittent access, as have others. We've dopamine sensitization, but we hadn't really looked at psychomotor sensitization. So that took us all the way back. So the question in this 2020 paper was simple. Does intermittent access produce psychomotor sensitization? And does it have the kinds of characteristics that we saw decades ago looking at experimentally administered drug? And so that was what Crystal did. And in one study, she compared long access to intermittent access and then looked at animals tested early after withdrawal, like I think it was 1 day after withdrawal versus 30 days after withdrawal. And what we saw was, indeed, if you looked at 1 day after withdrawal, we saw intermittent access resulted in psychomotor sensitization upon a drug challenge, whereas long access didn't. But if you looked at 30 days of withdrawal, now both long access and intermittent access animals showed psychomotor sensitization. And the degree of sensitization in the intermittent access for animals, for example, was greater after 30 days than it was after 1 day. And that's exactly the kind of thing you see with all of the studies, the early studies with experimenter-administered drugs. So that's one characteristic of psychomotor sensitization that matches the old experimenter-administered drug studies that same kind of thing is produced by intermittent access. Crystal also looked at sex differences. Intermittent access produced greater psychomotor sensitization in males versus females. Another hallmark of sensitization is cross-sensitization. Animals pre-treated with a drug like cocaine are sensitized to amphetamine later and vice versa. So Crystal asked, does intermittent access to cocaine also produce cross-sensitization, psychomotor sensitization to amphetamine? And it did. And so the main point of that paper was when you use intermittent access, you now see, again, all the hallmarks of sensitization that are consistent with the characteristics of sensitization that had been described decades earlier. And sort of, for me, it was sort of like closing the circle, and coming back and putting the final touches on the fact that indeed self-administered drug does produce psychomotor sensitization, incentive sensitization, dopamine sensitization, and it produces more addiction-like behavior than long access. And so, again, the main point there is that or the broader point is it really is important to consider these kinds of pharmacokinetic factors as well as pharmacodynamic factors in terms of the ability of drugs to change the brain. I mean, temporal pattern really matters in this particular example of experience-dependent plasticity, which is drug experience-dependent plasticity. And we know that for all kinds of other forms of brain plasticity. Just learning and memory. You have mass trials versus space trials. Time matters. LTP versus LTD. Little bits in terms of differences in temporal pattern matter in terms of how brain changes. And I think this intermittent versus long access story is an example of that. That is, the brain really cares about the temporal pattern by which stimuli, in this case drugs, impinge upon the nervous system. And that has a big effect on how the brain changes.
Interesting. So would you, so I guess I'm trying to get my head around what does the fact that these different types of models and their various nuances that you've, over many years been piecing apart, what do these sort of tell us about the disorder of addiction? Like, is it the case that, for example, the intermittent access model is a better model of addiction than long access? Or is it that we're learning different things from those different models and they're sort of like different aspects of addiction that we're studying or something? Or is it that, if we have a model that induces a phenomenon that we believe is theoretically linked to addiction, like incentive sensitization, as you guys have argued over the years, that whatever behavioral model gets us that kind of outcome is the one that's the best model of addiction, so to speak? Or is it that, we should be really using all these different models to get converging evidence, or is it, or something else? Like, what's your take on this kind of question?
Yeah, I think it's hard to talk about best model because it depends on what you're trying to model. To my mind, something like long access and intermittent access are modeling different kinds of things. So if you're interested in studying, for example, the withdrawal syndrome, that also can contribute to resumption of drug seeking and drug taking behavior. Something like long access produces a protracted withdrawal period. It lasts 1 or 2 weeks or so. And during that time, and I think one confusion in the literature is that during that time, sensitization is masked. And it's only when withdrawal lifts that you now see the expression of sensitization. And there's some very nice studies showing how indeed sensitization is there. It's just masked by whatever changes in the brain are producing tolerance of various drug effects. And so if you're interested in looking at that aspect of addiction, then long access, I think, is very good for modeling that. If you're looking, I think intermittent access is much better at looking at sort of how drugs might change the brain to increase incentive motivation for drug, even after long periods of abstinence. And I think it's reflecting the kinds of changes that you get that are related to, again, the persistence of addiction and so on. I mean, they both have implications in terms of theoretical models. I mean, intermittent access is very consistent with incentive sensitization, for example. And so we would argue that indeed, those kinds of studies are consistent with that kind of a view of addiction. But when you think of addiction in people, of course, what these animal models are doing, again, to my mind, for the most part, is they allow you to isolate how the drug itself might change the brain to influence, the development of addiction, which is very, very hard to do in people, because in people, what you're usually often doing in imaging studies, for example, is studying people that have taken drugs for 20 years. You can't, all of the changes in brain took place 20 years ago. And so looking at people after 20 years of drug use aren't very good to isolate, how drug might change the brain. And so the animal models, I think, are very, very useful to trying to get a handle on that. But again, the complexity of addiction in people, of course, is not captured at all by any so-called animal model of addiction. We're really looking at things such as drug-induced plasticity and how it might change brain to render. But when you go to humans, you got so many more complex interacting factors, social factors, as well as drug-induced biological factors and so on, which are, I mean, people do try to model these, of course, with enriched versus impoverished and so on, but you're looking at a narrow slice of what you think of as addiction. So, better model. Depends what question you're asking. And again, if you're interested in how drugs might change the brain to lead to enhanced motivation for drug for long periods of time, even long after withdrawal, I think intermittent access is capturing that much better than long access. But again, most of the studies with long access are looking when animals are off drug for relatively short periods of time, well, they're showing tolerance. If you look at long access animals 30 days out, they're sensitized like crazy. There are relatively few studies that look at long periods of withdrawal following intermittent access. So again,
Interesting.
depends on the
Yes.
question.
Yeah. So then arguably maybe to be a little provocative. So if somebody were to be given a bunch of drug for some period of time, let's say they're in the hospital and they get morphine or something for their pain for a while, and then they kind of go off the morphine, they may experience withdrawal, but they may not actually be addicted in a way that is kind of clinically relevant, where even though they are dependent physically on it. And so arguably these long access models may be then studying that phenomenon, which is an aspect of addiction, obviously, and does interact with some of these other things in people who voluntarily use drugs, but is in fact parsable both neurally, et cetera, and also sort of phenomenologically, from the process of what you would call insensitive sensitization, or that compulsive motivation to keep using and relapse and all that kind of thing, those aspects of addiction. Is that fair to say, or is that?
Yeah, no, I think that's fair to say. And it's also important to remember when people are given drugs therapeutically, like opiates, for example, you tend to want to bring your blood levels of drug up and maintain it at a relatively steady level. We know from animal studies, again, going back decades, that tends not to produce sensitization. Being exposed to drug intermittently is really critical in producing these sensitization-like effects. And that might account for why, and indeed, if you look at the data, people that are given things like opiates medically with relatively sustained levels like that, when they come out of the hospital, tend not to have problems. I mean, they may have some withdrawal, but they tend not to develop, compulsive patterns of drug seeking and drug taking behavior that you refer to as addiction. There's a very recent paper came out. I can't remember which it was, but looked at the incidence of problematic drug use in people with that had taken drug for medical reasons, and it was very, very, very low. And that could relate, in our mind, to what the fact that they never had drug in a pattern, in a pharmacokinetic, pattern that actually produces really robust sensitization,
Interesting.
which we would argue is really necessary to produce this, sort of compulsive patterns of drug seeking and drug taking behavior.
Huh. So what's your take then on I mean, there's this sort of story out there these days with obviously with the opioid crisis that we're experiencing as a country, continent, for sure, that has to do with the availability of opioids. And so there was this, sort of pharma companies were pushing their new opioids, which they claimed were doing what you're saying, which is creating these steady state opioid levels that are not going to be addictive for all the reasons you're just mentioning. And yet somehow this is linked to this crisis, it seems, in terms of people, starting to use these drugs, perhaps medically at first. But would you then say that it's not that medical use that's the problem? It's that when they divert it and maybe crush up the oxycodone and snort it and get that kinetics that's more relevant to your intermittent access kind of situation, that's when the addiction comes into play. It's simply just availability, and that then leaks into some sort of abuse-like patterns in some people because they have their hands on the drugs. It's not so much that they, got the drug for their, you know,
Yeah.
injury or something like that, and then now they just sort of go down this downward spiral, so to speak.
Yeah. I think that's highly likely.
Okay. And then another question I had, is obviously a lot of the work that you've done over the years. I'd say most of the work, as you mentioned, also was with psychostimulants, especially cocaine. And then obviously there's other kind of competing models of addiction that are out there where people are emphasizing more of these, decreases in dopamine that can be kind of self-medicated by the drug and withdrawal and things like that. And I don't think it's a coincidence that most of the groups that are coming forward with those theories, George Koob in particular, are mostly studying other kinds of drugs, opioids, alcohol, that have more severe withdrawal symptoms than do these psychostimulants. How does that play out in your mind in the concept and conceptualization of addiction, and how these competing theories sort of explain different aspects of the disorder? Does drug matter, or is it really just, I should also say, as you obviously know, heroin and alcohol and things do also cause psychomotor sensitization if given under the correct protocols. So is it sort of that that everything is really just comes down to these basic incentive motivational processes that are sensitized, for example, or is it that there's something drug specific and that like an opioid addiction is actually different than a cocaine addiction, for example, in the brain?
Yeah, no, a question like, is an opioid addiction the same as a cocaine addiction, is a pretty complex question to which I would say no one knows the answer. I think there are going to be similarities and there's going to be differences. But in terms of thinking of sensitization as a process, it's, again, there's lots of studies, and some of the earliest studies on going back on psychomotor sensitization were with opioids, showing that repeated intermittent exposure to opioids produces robust sensitization. Looking at opioids can be tricky at looking at something like psychomotor sensitization, because opioids tend to produce, depression of motor activity. And so you're always, having to set things up so that you can see the, psychomotor activating effects of opioids, over and above their motor depressant effects. One way Paul Vezina did that, again, decades and decades ago, was to put opioids directly into the VTA. What you get then is what looks like amphetamine. The animals show great psychomotor activating effects, and they show, sensitization, because you don't see these motor depressant effects. Some strains of mice with opioids show primarily, psychomotor activating effects and not much in terms of motor depressant effects. So you can really see it much more clearly that way. So yes, so it's trickier looking at these kinds of, particularly psychomotor effects with things like opioids, but there's a large literature looking at opioids and alcohol and many other drugs that do produce sensitization. One thing that it's curious, and it's not looked at that much in the opioid field, and I think people should do it, is, an opioid is not an opioid is not an opioid. If you look at just sort of the influence of morphine versus heroin versus a drug like remifentanil or fentanyl on dopamine, there's a huge differences in terms of different opioids, all being opioid receptor agonists, but there's huge effects in terms of the degree to which they activate dopamine systems. Morphine is relatively ineffective, heroin more, but when you get into fentanyl, remifentanil and stuff, they look like amphetamine. They produce huge dopamine surges. And so never mind comparing opioids with psychomotor stimulants, I think there should be more comparison between opioids, trying to figure out what's going on in terms of different opioid actions on things like dopamine signaling and the ability to produce things like sensitization, tolerance, withdrawal, and so on. Because the opioids are very, very different, just, remarkably different. And best I can tell, it's not something that's really studied or emphasized that much.
Yeah, I would totally agree. And yeah, that's certainly my impression as well. And it's obviously a very relevant point right now, given that, for other reasons, legal and sort of societal reasons and things, fentanyl has kind of taken over
Fentanyl's
a
what's
lot of
taken off, yeah.
the drug
With
supply these days. And
much faster kinetics.
yeah, which is implication. I mean, the kinds of implications I usually hear about are, heroin, for example, is a longer lasting drug. And so the physical withdrawal is going to kick in later. So, for example, a person who's addicted to heroin may take some before they go to bed and wake up, not necessarily in severe withdrawal, whereas fentanyl, that's probably not the case. And so that right there can, to the extent withdrawal is motivating, avoidance of withdrawal is motivating their use that day. That's going to have, some particular implications. It's not just the potency, sort of it's smaller, it's easier to put in a sneak across the border or something like that. There may also be these other factors that are really important. And of course, how they influence with reward systems is another huge one, as you mentioned. And then kind of relevant to the same general topic, uh, setting aside whether there's sort of different, whether addiction's a different thing for opioids or cocaine, even within like a drug, um, would you say that, for example, there's a different phenomenon that we call addiction, that in terms of how it works in the brain, that kind of thing, in a male and a female, animal, human, and what about these hormonal changes that you've been studying for dozens of years? There's obviously hormonal changes across the day, across the month, across the lifespan when you're thinking about puberty and menopause and other kinds of hormonal changes that come with age. Do you have a sense that this is sort of essentially just like tweaks on the same phenomenon that basically exists, that is addiction? Maybe it's incentive sensitization, in all cases, and it's kind of tweaked here and there by hormones and sex and things? Or is it really just that there's like two different phenomena that are in the two sexes, or that there are two, multiple different phenomena that have to do with hormone interactions at these kinds of timescales?
Yeah, I don't think anybody really knows. Um, I mean, there's some differences that are probably just sex differences that are probably quantitative, but there very well could be other differences in terms of which brain systems and circuits are changed in exactly which way that could be qualitative. And I think that's something that a lot of people are looking at now. I mean, the tools available now to try to tease that kind of stuff apart are much more developed than the tools that were available, back in the old days. But it is certainly the case when you start to think about sensitization, incentive sensitization, and so on, that there's a huge number of factors that interact to determine when you take drug now, at this particular point in time, whether that drug is going to change your brain or not. And those factors include things like the context that you're in. Are you under stress or not? What hormonal state are you in? Are you a male or a female? Is it a drug-associated context? All of these kinds of factors are interacting in very complex ways, which we don't understand at all, but they're interacting in very complex ways to determine whether at this point in time, how susceptible your brain is to drug-induced plasticity, such as sensitization. And we don't have a really good handle on how these interactions, we know a fair bit about each of them separately, but how they interact which there always are, of course, particularly in people. How they interact is not, it's just not well understood, but they are interacting. So
Yeah,
you're,
it makes it. I
go
was going
ahead.
to say it makes it even more complex when, even within one of those subcategories, let's say a female in this estrus phase, and this kind of kinetics of the drug that you're giving them, you still have individual differences even within them, with not necessarily explained by these overt sort of dichotomous kind of variables. For example, the sign and goal tracking individual difference that you and Shelley Flagel and others have studied quite a lot over the years. There's something perhaps different about these kinds of animals that tend to attribute certain types of salience to this versus that cue, that also seems to impact downstream susceptibility to different factors to do with addiction. Would you like to comment on that?
Well, yeah, there are, again, we've known for decades and decades in terms of sensitization that there's enormous individual differences. Some animals are highly susceptible, some animals are very resistant to sensitization. And again, we've known that for a very, very long time. We know a reasonable amount about the factors that confer susceptibility in terms of, and again, sex, gonadal hormones, context, environmental factors, and so on and so on. And the sign tracker, goal tracker story is actually fairly complicated when it comes to those individual differences. I don't know if we want to get into that story. But yes, individual differences in terms of susceptibility to drug-induced plasticity, such as sensitization, are the hallmark. And those individual differences, and I think the sign tracker, goal tracker model is a very good one, can provide really good insights in terms of what's going on, what systems are necessary and sufficient to induce changes that lead to changes in incentive motivation and so on. And the sign tracker, goal tracker model has been very, very good for teasing apart how brain changes in different animals to render cues that have been associated with rewards, with a lot of incentive value versus just with predictive value. And it allows you to tease apart things like motivational processes from learning processes. And Shelley's making, a lot of strides using that model to ask those kinds of questions. And lots of other people are getting interested in it now too.
Sure. Yeah, and then that actually brings me right to another topic I wanted to ask you about. So when I first started doing this series, it was in part motivated to discuss the one of the more highly cited papers in Psychopharm, which was by your longtime collaborator Kent Berridge, discussing the sort of state of the field at that time in 2000, what was it, 2004 or something like that. Of what is the role of dopamine and reward? And there's this at that time there was I think the original prevailing hypothesis of the role of dopamine reward in that coming from Roy Wise, which is that it kind of mediates pleasure basically from all kinds of different rewards. And that's sort of why since drugs activate dopamine, they are pleasurable and that's why they're addictive. And I think that theory had kind of started to fade at that point, but the controversy about the role of dopamine in kind of motivated behavior, motivated processes like incentive motivation, versus learning, as especially exemplified by a lot of the reward prediction error stuff that was coming out at that time from Wolfram Schultz and many others. That controversy, I think, to some extent, still is raging. I was wondering if you can comment a little bit about kind of your take on the role of dopamine in motivation versus learning. From your own work, from the conceptual work that you've done with Kent Berridge, and then some of the stuff that's happened in the last 20 or so years. Has that changed any of your conceptualizations of the role of dopamine in these processes? And maybe you can just comment a little bit about how you think about that these days.
Yeah, the first comment I'll make is in terms of controversies associated with the role of dopamine reward. Going back to the early days, as you said, the primary hypothesis back in the 80s was Roy Wise's idea that dopamine was the brain's pleasure transmitter, and the sort of called anhedonia hypothesis of a neuroleptic function. I should mention, I said during the 1980s, I probably published 20 papers on sensitization and never mentioned addiction. And part of the reason for that is that the meme was so strong at the time that dopamine mediated pleasure, so if dopamine systems sensitized, that would mean addicts got more and more pleasure, and that doesn't sound like addiction to anybody. And it was partly due to sort of how strongly ingrained in the literature that idea was, that we never really made the connection in these early sensitization studies between dopamine and addiction. It was only with the idea that came out of Kent's work was, well, yeah, dopamine doesn't mediate pleasure. Dopamine seems to be more involved in mediating motivation for drugs, the extent to which they're wanted. And Kent coined the phrase wanting in quotation marks. That led to, well, when you take drugs repeatedly and the system sensitizes, that would mean they actually want drugs more and more and more. That sort of sounds like addiction. And it was that led, just that core of an idea that led to the idea or the foundation for incentive sensitization theory. Now, dopamine, of course, getting in, what does dopamine do? Dopamine does lots of things. I mean, dopamine does all kinds of things. And all of the stuff and Kent writes much more in this than I do, but the stuff that Kent has written about and the things that I've done with Kent, we always make clear, we're talking about the role of dopamine in reward. What's the role in reward? And it doesn't mean that that's all dopamine does. It doesn't mean that there are not dopamine cells and circuits that do many other things. They do. They clearly do. But in terms of asking the specific question, what is its role in reward, reward-related behaviors? I think the idea that dopamine's role in reward is to mediate the extent to which they're liked is pretty much settled. Although you'll still see it come up, even now, sometimes people sort of slip into that thinking.
Especially in the popular media and things like that, but even in the
Certainly in
scientific
the popular
literature.
media, actually, yeah, but it still comes up. Yeah, so but the bigger, as you said, the bigger ongoing controversy has to do with the role of dopamine in learning versus motivational processes. Not to say it can't be involved in both of those as well, but you can ask about the extent to which the role of dopamine in reward is associated with its influence on learning versus motivational processes. And Kent has written extensively on that and made lots of very cogent arguments as to why he thinks that the role of dopamine reward is much more related to motivational processes than to learning per se, which by learning I mean the formation of associations between, say, a CS and a US, or an action and an outcome, and so on. And so that is an ongoing controversy. One study that we did on that, relatively recently, that I think it gets ignored in terms of some of these arguments, was a study that Ben Saunders did when he was in the lab, that was published, I think, around 2012 in the European Journal of Neuroscience. And this was a sign tracker goal tracker study. And it involved looking at the expression of sign tracking versus goal tracking following dopamine blockade by injecting flupentixol into the nucleus accumbens. And there was a follow-up on studies showing that if you block dopamine, you block the learning. These were studies by Shelley. But if you block dopamine, you prevent the acquisition of a sign tracking CR, but not of a goal tracking CR. And what Ben was looking at was, well, after you've acquired a sign tracking or goal tracking CR, does dopamine blockade influence the expression? And what he found was consistent with the acquisition data. That is, dopamine blockade completely attenuated the expression of sign tracking CR. It had absolutely no effect whatsoever on goal tracking. Now, that alone gets into the controversy about the role of dopamine in learning versus motivation, because goal tracking is the classical Pavlovian conditioned anticipatory approach response on which the ideas of things like reward prediction error came from. So why the hell is goal tracking not dopamine dependent if it's being mediated by something like an RPE? Now, and also, of course, Shelley's earlier studies showed that you don't get the transfer of the dopamine signal from the US to the CS in goal trackers, although you do in sign trackers. Okay, so now you can start to tell some stories. Well, goal tracking, maybe it's more akin to a much more cognitive kind of learning, where the animals have sort of an explicit expectation of a reward, which would be sort of akin to act outcome in the instrumental world. And we know that that's probably not dopamine dependent. So that's why goal tracking is not dopamine dependent, but it looks like sign tracking is. Well, first thing you have to do for people that want to talk about dopamine as an RPE kind of signal is, so which kind of learning are you talking about? Is it all kinds of learning? That gets very vague. And why not goal tracking? But the other thing Ben did was to show, well, yes, so sign tracking looks dopamine dependent, but even in the sign tracking, the learning is not dopamine dependent. And he did two additional things. Yes, sign tracking as indexed by approach into close proximity with the lever in those sign trackers, that's degraded with dopamine blockade. But if you look at a different condition response, when you train animals you only look at one condition response usually, but they're producing many condition responses. And so another condition response we looked at that you see in sign trackers and goal trackers is condition orientation. When the lever CS comes out, the animals orient to it. Sign trackers then approach into close proximity to it. Goal trackers orient to it and then go over to the goal cup. But that orienting response is a conditioned response. You can show that you see it in paired animals, but not unpaired animals, and so on. After dopamine blockade, that conditioned orienting response is completely intact. They don't show the sign tracking response. They don't approach the CS into close proximity with it, but they still show condition orienting to it. That tells you the CS-US association is intact. The other thing that Ben did was when he put the flupenthixol in the accumbens prior to a test trial, he looked at them at the very, very first trial on the test. Well, no new learning can have taken place on that very first trial, but their behavior, that is the sign tracking behavior, was completely down. That effect of dopamine blockade can't be on learning because there's no learning taking place there, which is consistent with the condition orienting kind of response. And that was what led us to suggest that, listen, the effect of dopamine blockade is on performance. It's not on learning. The learning, the CS-US association is there, but the animals don't perform the sign tracking CR because the CS has to have incentive value to draw the animal to it. This is Kent's motivational magnet property. What you've done is degraded that. So they don't do that, but you can see that the association's fully intact. And so if you're going to think about dopamine as a reward prediction error, you have to be able to explain phenomena like that. And I always even in undergraduate lectures, I always try to remind people, you can't measure learning. All you can measure is performance. Learning is an inferred psychological process. You infer learning by changes in performance, but lots of things can influence performance besides learning. If you do brain manipulations, you can influence what looks like the learned response, yes, by, say, degrading a CS-US association, or by influencing attentional processes, or influencing motivational processes, or influencing motor processes. There's all kinds of other things that can influence the learned behavior besides having an influence on learning per se. And I think that's this paper of Ben's is a good example of that. That is, the effect actually isn't on the learning, even though it looks like it is, it's on the performance. And the learning literature is full of examples like this, where people think that they have an effect on learning. No, but it's actually on some performance variable.
Yeah, this is where it becomes, I think you would agree that thinking about these things in the psychological way that I think we both do, really kind of deepens the story, both in terms of it makes it a lot more complicated, but it is possible if you carefully watch animals and you design experiments well to start to tease these things apart. And that's something that you, I think, have really done over the years and have contributed to a huge extent to the sophistication of the field on these kinds of topics to its benefit, I would say. And so I guess with that, we're about out of time, but I just wanted to thank you again, Dr. Robinson, for taking the time to have this discussion. I found it really fascinating, and you've been a huge influence in my own career, ranging from when I was doing master's degree work in Harriet DeWitt's lab, looking at dopamine and smoking craving. I was reading your papers. In fact, I think I told you Harriet gave me a individually personalized sort of copy of a reprint of one of your early review papers on this topic that you sent to her at that time physically in the mail, and she handed to me, and that I still have that paper. It's been really influential, and obviously was a huge part of why I went to Michigan and had you on my committee and stuff. So thank you also for the personal aspects of our relationship over the years. Have been a tremendous influence on me as a scientist, and I know that I'm not the only one that can say that. So thank you again, Dr. Robinson. I hope this has been a pleasurable conversation for you and also for the listeners.
Yeah, no, I really appreciate the opportunity to chat. It was fun. I like your comment, physically in the mail. There was a time, and that's the only way we could communicate.
Right. Yeah,
As
those things are a
physically
little
in
bit lost
denial.
now. I probably wouldn't have the PDF that you email Harry or
Yeah,
something,
right.
so
Times
some of the
have
advantages
changed.
of modern life have downsides too, but so it
It
goes.
was great talking with you.
Nice talking to you too. Thanks again.
Okay, take care, Steve.